Welcome, everyone. We're going to get started here with the next panel, which is the hottest new ideas in oncology. My name is Farah Gerdes. I am a partner at Wilson Sonsini, Goodrich & Rosati in our Boston office, where I lead our technology transactions practice. And I'm going to start off by letting our panel introduce themselves. So, Brian, if you'd like to kick us off, that would be great.
Hi, I'm Brian Doherty. I work at AstraZeneca in translational research in the oncology R&D.
Hi, I'm Roy Herbst. I'm here at Yale. I'm the Deputy Director of the Cancer Center, and I also work with the Dean's Office on Translational Research and Industry Alliances, so very happy to be here today.
I'm Fred Cohen. I'm actually here for my 45th college reunion, so gave me an excuse to be here. I'm a physician scientist who got turned into a business guy about 20 years ago, and I now am the chief investment officer for a group called Monograph Capital Partners.
Hi, I'm Teresa Vitetti. I'm the president of Takeda's global oncology business, and so I've been working in oncology for the last 15 years.
Hi, everyone. I'm Eugenie Scarlett. I'm a principal at MPM Capital. We have a lot of exposure to oncology and have a background in cancer immunology, so oncology is near and dear to my heart.
So we have an impressive panel with investment, company, and academic perspectives. So we wanted to kick it off just to start with a little bit of the science, and maybe I'll turn it over to Roy to talk about some of the innovations that you're seeing today and what's exciting to you in oncology research.
Thanks so much, Farah, and really delighted to be here. It's amazing what's happening in oncology today. I think many of you know that, but let me just recap a few things that are on the top of my mind. You know, we're in an era now where people come to a major center like Yale or any cancer center, and it's expected that you'll profile their tumor from head to toe, and we'll know everything about the genes in the tumor. And now I think soon it'll be expected to profile the genome as well and know about their normal genes. And really the mantra is, you know, the right drug for the right patient at the right time. So we're really in an era of precision medicine. And we're seeing that. We see that in so many cancers. The area that I work in, lung cancer, if someone has lung cancer now, it's a question of do they have a driver mutation? And if they do, can we treat them? Of course, there are 8 or 9 different oral agents we can use. The problem, of course, is resistance. So we have to target resistance. Same in breast cancer, CML, lymphoma. So really, it's the personalized and precision medicine. That's wonderful. Of course, the problem is not every driver is actually going to shrink tumors, and we learn that there are many resistance pathways. Then there's the immune revolution. A lot of that work done here at Yale, work around the world. The fact that we can actually harness the immune system to target a tumor, it's phenomenal. I'm heading to the ASCO meeting just in a few hours, and half the meeting will be on immune therapy. 20 years ago or 10 years ago, people would have said, you can never use immunotherapy. In fact, panels like this would have said, vaccines are not going to work, immune therapy is not going to work. But we are channeling the immune system to, in many cases, I would say cure cancers like melanoma, renal cancer, lung cancer. The immunotherapy works pretty well in a lot of other tumors, but again, we've hit an area now where we have to understand the mechanisms, resistance, tumor microenvironment, but amazing potential to do that. And of course, there's big data, AI. We talked about this. There was a panel on this very stage yesterday. I think AI is going to be so important as we take all these data, clinical trial data, molecular data, and that can help us to identify who benefits more, who doesn't. That will be important both in terms of more effective treatments, less toxicity, and also from an economic point of view. And then one thing I just feel I should mention is also access. I think one of the most important things about oncology now is we need to get these drugs accessible, and that's going to mean being more pragmatic in how we do our trials, getting drugs that are more available, are easy to administer. But with all that said, it really is a time where the entire community, academic, industry, patients, the NCI, the FDA have really come together, and we are making progress, but there's still much more to do. Thanks.
Brian, do you want to respond to that from the company perspective on the R&D side and what you're seeing going through companies and the promise of precision medicine and some of the other innovation that, maybe this way to Brian?
I wasn't sure.
Or Teresa.
The name, it's weird.
What you're seeing in companies in terms of the R&D and the focus there.
Roy just captured it really well, right? I mean, I look at, I reflect on Takeda, which is the company that I'm running our oncology business. We're very focused on the innate immune system. So with immunology, there was been a lot out there in the adaptive. Now there's a lot of work around what can we do with the innate system to enhance that, to make that more effective. And on the precision side, as you said, the more and more we're able to sort of with genomic sequencing, with testing, that's where the focus is right now. And it's interesting as you look at what you said, the late breakers, the big news coming out of ASCO. It's those themes of the immune activation, it's the precision, the targeted therapies, it's the ADCs that are now sort of the hot item as we look at late breakers, et cetera. So, I mean, that's where pharma's focused, is in exactly the places that Roy just detailed.
So then if we look at the investment side, and maybe turning to you, Shani and Fred, from a lawyer's perspective, a startup company's perspective, the financing environment is becoming harder and harder in the current economic environment. I think everybody would probably be interested in hearing what you're focusing on investing in, what's looking riskier or more appealing to you, and how you're focusing your dollars.
Yeah, great question. I think whenever we look at 2023, 2022, 2021, we go, wow, we're in this really interesting time relative to, the peak, which was in 2020, 2021. I think there's a course correction that's happening right now, just from an investment standpoint. If you zoom out and you have that macro lens and you look at the last 10 years, year over year or quarter over quarter investment within the sector, I think it was a nice slope up to 2019, and it was really the pandemic era that really drove that spike. So I think the numbers were 12 billion a quarter was coming into the sector at the peak of that spike, which was in 2021. Whereas in 2018, 2019, it was single-digit billions of dollars coming into the sector. And so a lot of that really had to do with reallocation of funds that groups that typically don't invest in biotech had and wanted to deploy. And at the time, biotech, life sciences was a very appealing area. And so we had a massive influx of tourist investors. I think what I'm saying is not necessarily new. I think this is something that we've broadly commented on over a number of years in the last maybe 2 to 3 years. And so there's a course correction that's happening in 2023. There's still a lot of capital to deploy. There's still a lot of funds raising new funds. I think only just this year, there were 3 new funds that were announced specifically for therapeutics or biotech. So capital is available. I think the difference is there's just more discipline with how capital is being deployed. There's also maybe a bit more discipline in how we think about valuation. So we're now in a buyer's market. And I would say valuations are starting to come down in the private side. We saw valuations coming down quite massively on the public side last year and maybe towards the beginning of this year. We're now seeing that on the private side. So we're now seeing Series Bs kind of going back to what they were pre-pandemic. So there's this course correction that's happening on the valuation side and a bit more discipline in how management teams are actually deploying capital around their portfolio. So again, there was this overexuberance that was happening during the pandemic, maybe less discipline in how capital is being deployed, and maybe just an overexuberance in regards to valuation expectation. So we're coming back to somewhat of a normalcy, I suppose. And what that means is, there's a number of companies that are still very early, preclinical stage, and have these massive valuations that it's still going to be hard for them to raise money or new financing rounds at the valuation at the price that they set in the prior round. And so for us, I think we're still trying to ensure that we're consistent with what we've always done, making sure that the price really fits kind of the stage of the company and the stage of the opportunity. When you think about certain spaces, I think the prior panel mentioned cell therapy. That's really expensive investment opportunity or space to invest in. And so when you think about the valuation expectation going into a space that's going to require hundreds of millions to actually get to that first meaningful data card, you really have to do that calculation and really say, okay, what is the stage and how do we actually think about deploying, making investment within those specific technologies.
And Fred, maybe you can provide your perspective on the sorts of technologies that are interesting to you or the stage of asset and the risk factors that you're particularly focused toward.
Well, our read on pharma is today they're very interested in phase 2 assets, but they're much less interested in platforms. They've got about $120 billion of annual sales that will come off patent between now and 2030, and that's a lot of potholes to fill. So what they are looking to biotech for is probably 70% of the hole fillers. That's just what the statistics say. When we look at oncology companies, we look at the science, we look at the medical rationale, and then we look at how practical it is to get from where you are to where you need to go. And it's that pragmatic part of it where many things fail, because to Roy's point, there's a lot of very interesting science. There are a lot of important medical opportunities that are underserved in oncology. As one of the areas that I think a number of people have been looking at now is the area of ADCs, because antibodies clearly work, but antibodies armed in a certain way work. I would say that radiopharmaceuticals are an example effectively of ADCs. We've been spending some time recently on radiopharmaceuticals, and they're looking at alpha emitters and then trying to figure out how the hell are we going to get enough actinium for all these companies, and or is it going to be lead? And I don't think it's going to be astatine. So there are all these pragmatic aspects of drug discovery and development. We've looked at a number of cell therapy opportunities. I've been involved in a bunch of cell therapy companies. The ones that have made real successes, it's unbelievably impressive. But now when you look at some of the cell therapy ideas with various T-cell receptors and HLA restrictions, actually finding the patients to do the trials is super hard. And so there are pragmatic aspects, and so it encourages us often to work backwards from the endpoint. Okay, this is what I need to prove. How likely is it that I can actually do that with some rational amount of money and some rational amount of time?
I'll add on to that.
Yep.
Is this mic working?
Yes.
Sitting on the pharma side of things, it is true. I think that there is much more of a need to make sure that your pipeline is filled with the later stage clinical assets. And this is where larger companies, big pharma, can bring resources to bear to bring that forward. Although, not to give up hope, there is definitely interest in the platforms and things there, right? Because I think that there would be too much hubris on the part of big pharma to think that you can get all of the science out of your own labs. So there continues to be interest, but I do think that the rigor in which one has to sort of look at how you're deploying the resources in the early phase needs to be more careful. Or you need to figure out, we had done work with, which we now, had purchased, was Gamma Delta and Adaptate. What are the interesting ways where you can invest, where you can partner, and then have an option to purchase? So there's a lot of different ways, I think, in which to access it. But you're right, big pharma does remain highly interested in the clinical stage assets.
You look like you want to jump in, Roy.
No, I was just going to emphasize what Fred's saying. It's become super personalized now. So the cell therapies, are we going to be able to make a vaccine or a cell therapy for any given patient? The ADCs, it's incredible what they can do, and the different payloads, and who would have thought they're really chemotherapy, but being delivered in a much more precise way. But that's going to mean we need the tissue to find the target, and there's only so much tissue as a clinician I can tell you get from a patient. So we're going to have to find ways that the whole medical community can come together. And on that first biopsy, we analyze it for 5, 6, 7 targets. So I'm going to throw out here that it's going to require a little bit of pre-competitive collaboration and some regulation, dare I say it here, but to try to bring together ways that at that first biopsy, we have to make sure we get all this information. And actually, not just from the patients that can make it to Yale or Harvard or wherever, but from the patients in the community, the people from New Haven and people from the community and the diverse populations, which are very much underserved in this because they don't even make it to the first step.
Yeah, just to add on to that, yeah, Roy, good point. Tissue's the issue, right? No samples, no science, as they say in our department. If we want to do precision medicine, we need tissue, and it's very small amounts. We're incorporating a lot of diversity in that sampling that we're doing within our company, and I think the points about ADCs in particular and radioimmunoconjugates, these are ways of targeting chemotherapy and radiation. So it's going to be a next generation approach. There's a lot of complexity around delivery to the target, just classic ADME things as well, and how hard you hit the target, and then layer on precision medicine for the human variation. It's really challenging, but there's a lot of excitement in the fields in particular. I think you're going to hear a lot at ASCO, where Roy mentioned that he was going, and that's really where the rubber meets the road, because you're bringing in the clinical data and seeing if all these great ideas are actually benefiting patients, and that's really what drives the decisions in oncology in particular. Is where's the unmet medical need, right? And then we start figuring out how to fill in those holes in those potholes in the pipeline.
So I think some of that's a good transition to the next topic. We're going to talk about both partnerships with academia and then partnerships between pharma and biotechs. Maybe to hit the first part of that first. Brian, I'd be interested to hear your perspective on partnering with academia. Part of our pre-discussion among this group included investors looking for assets to stay potentially in academia longer, so that there was more potential for initial investments to get assets into the clinic, so that they were well positioned for Series B investment. And part of that included partnering longer with academic institutions that have access to different forms of capital. We see a lot of investors sort of serially going back to individual institutions and investing in technologies that are coming out of those institutions. How is AZ partnering with academia, or can you provide some of your thoughts there?
With academia, it's pretty diverse. At our company specifically, we have a couple of examples of partners of choice where we've kind of worked out with leading oncology institutions around the world how we can get the paperwork in place to begin to do these collaborations, everything from tissues to technologies to potentially therapeutics. There's innovative approaches being taken, say, at our Gothenburg, Sweden site, where they've actually set up an incubator where there's 30 different companies in small biotechs, as well as some academic groups, all working within the ecosystem where the big pharma is. And that model's been deployed to some of our other sites as well, bringing in incubator startup companies, reaching out to academic groups, and also bringing in larger flagship biotech platform and therapeutic companies.
Teresa, do you want to share any of your experience there?
Well, we actually do a lot with academic institutions, because they have the science, they have the agility to sort of move at speed and develop things. We have the funding. And then at some point, we can bring it more into our development machinery, which is, it takes a bit of a lift to get things through, things to the FDA. Really? But, even, and a little shout out to Yale. I'm sitting on the board of Osmol Therapeutics, and look at the science that's coming out of Yale University. And being able to pull that forward as well, I think, is really important. So it goes both ways, I think, in terms of the synergy between what you can do with academia, what you can also get from biotechs in terms of how you partner.
Yeah, just to add on to the New Haven area as well, as we're remiss to mention that Roy and I were on a meeting last week. There's AZ Oncology has a collaboration with Yale School of Medicine that's been going on for many years. And then I guess the other example you see is Alexion, the sponsor for this right down the street, a great example beyond academia, something that came out of academia, becomes a biotech that then gets integrated into a larger pharma. And even Alexion then goes out and acquires companies like Logic Bio for various technologies for delivery of medicines.
Yeah, from the academic point of view, it's wonderful, because we're running the large trials and bringing drugs to the community, but it's resistance, it's understanding mechanism and the science that's going to drive the next generation of drugs. By partnering with a company, we have access to tool compounds. Investigators can get together and interact. For the company, there's talent acquisition. They have people, sometimes people even leave and go work at the company, shared postdocs. This is all a wonderful way to sort of partner and come together. And I'll tell you, looking at the news over the last 24 hours, there's not going to be much more money at the National Institute of Health. In fact, there'll probably be less. So from an academic point of view, we have the science, we have the people, we have the patients, but we can partner in this sort of way, and I think it's huge. And then it can even go one step further, and we can partner across the nation as we put drugs to these personalized therapies.
What about from the investor perspective? When you see technologies coming out of universities, are you investing at that point? Are you looking for them to be more developed before investing in the companies that take those technologies forward?
So the question we often ask ourselves is, with this round of money, what are you going to do and who's going to be excited about that? Because the notion that, well, with this plus the next round and the next round, I will show you X, most people today are saying, I'll wait for the next bus to come. I'll see you in 2 years, I'll see you in 4 years. So the market today is one that incentivizes a number of people to look for inflection points. Those inflection points can be different. If you are looking at a particular enzyme inhibitor that is relevant in cancer, but the real issue of the existing agents is pancreatic toxicity, and it shows up in a monkey model, and you can show that your compound is clean there, that may be the value-creating event. Frequently in oncology, the value-creating event is phase 2A data, where you see efficacy. You're less worried about side effects in oncology. You can contrast that to investing in a company going after NASH, where what you really care about is phase 3 data, or in obesity, where it's the phase 3 toxicity that can make or break a compound. In most cancer chemotherapeutic settings, if you have a meaningful impact on pancreatic cancer, everybody's going to figure out how to deal with the side effects. So we're often looking for that aha piece that says, great, this now is valuable. In a number of so-called cold tumors, it would be an IO agent that could make them hotter. And there are obviously a number of cold tumors, and you'd love to be able to do something there. If you don't understand the mechanism, you'll never figure that out. In cell therapy, nobody really needs another CD19 CAR for a B-cell malignancy, but maybe for autoimmune diseases, there might be something there. Everybody's really interested is, can you do something in a solid tumor other than synovial sarcoma? So, you just have to say, what's the real question that people are going to care about? People, right? Like Teresa.
Right.
Right, because if you answer that question, it's going to work out. If you have one pharmaceutical partner that's interested, it usually ends up being a partnership. If there are 2 or more that are interested, you usually end up selling the company. That's kind of the rules we live by.
Yeah, I mean, at NPM, we have a fund that's dedicated to an academic relationship. So in 2018, 2017, we launched an innovation fund. It was $100 million exclusively to work with Dana-Farber PIs and a lot of the IP coming out of Dana-Farber. So what that really allowed us, NPM, to do is, one, be the first point of contact for investigators to share their new idea, their new breakthroughs. So oftentimes when, before a PI is ready to file their patent, they actually come to NPM and say, hey, can we get some guidance on, what are the next set of milestones we need to achieve? How should we think about filing IP? And we work very collaboratively with the venture group as well as with the TTO. So we are, I would say, a true partner, so to speak, with Dana-Farber. But we also have very strong ties with other institutions, either through the TTO or through their venture group or their innovation group. And because 2/3 of what we do at NPM is company creation, the relationships that we have with academia is critical to our business. And so we maintain those relationships, not only from, liaising with the PI, so to speak, around their technology, but oftentimes we may have a company that we're building because we're actively building companies as well. And even though the IP might not come from a specific lab, that PI might have specific expertise that's critical to really guiding the development of an early stage program. So we rely very heavily on academia for IP, for expertise, from a scientific or clinical advisory standpoint. So one of the companies that I'm working on right now that we're building, the IP is actually being spun out of a company, but we're looking to really build a team of advisors from 3 different academic institutions to really drive to that first value inflection, as Fred put it, in NHP models. And so for us, we always look to academia. And so I think you made a point, Farah, in regards to how early things are coming out of academia. I think last year there were 2 projects I was working on with 2 PIs, and it was very competitive. These are still very early technologies. In one instance, a target was not validated. In the other instance, we barely had hits for the platform. It was a small molecule platform. There were barely hits, and the hits were still just started to be validated. And the PIs basically said, if we don't get a term sheet in the next month, I have 5 other groups who want to put 10 million into this. So it was a very competitive market. We saw things coming out of academia maybe earlier than they should. Whereas this year, it's a really interesting pivot because another piece of IP, or maybe a company that I'm looking at, they have a program that's in IND enabling studies. And it's still within academia. So again, it's that course correction where we're seeing maybe less competitors coming into this earlier space, which is for company builders, kind of the relationships that we've fostered over the years that really allow us to get those insights early on. We're seeing less competition for those, and we're seeing a little bit more patience on the academic side to say, okay, let me take your guidance, NPM, let me just focus on getting to that next value inflection before building a company.
So a lot of what I'm hearing is that the key ingredient here is what Teresa and Brian want. So we want to hear from Teresa and Brian about what they want. What I've been seeing in the partnering market, because that's the core of my practice, is that has slowed down as well. There still are big deals that are happening. There are maybe fewer of them, but we're seeing things that are phase 3 asset deals that are big, ADC deals, as you've mentioned, CAR T deals, that are the big biobucks sort of deals that people look to. We're also seeing some of the AI platform deals, target discovery, compound discovery. And then in the prior panel, LNPs came up and different delivery technologies for mRNA products and new technologies along those lines. So maybe Teresa, to start with you, what sorts of things are you looking for to continue to build out your pipeline? What stage of asset are you looking for? Some of those elements.
Yeah, I think that, for us, we have cell therapy being a very hot area. And we did have a partnership and do with MD Anderson on NK cell therapy, right? So that's one area we're continuing to build out, trying to look at an allogeneic approach. Because it's from a commercial perspective, and you see it across the board, with the cell therapies that are marketed today, and they are good, they're good drugs, but it's really hard to get them to patients fast enough. The commercialization element of it and the supply element of it is a huge barrier. So I think that cell therapies is an area we continue to look for. We also look for places where we have the commercial muscle, we have the distribution, we have the development, so that if we can bring in an asset that is in the later stage, like we just brought in a colorectal cancer medicine that was, basically all along the path of development, we can commercialize that. That's important to us. Like hitting that unmet need, which is critically important. A lot of companies can't do that. So I think that it really comes down to, again, the things I mentioned earlier, where we're allogeneic cell therapies, the innate immune system. That's where we're focused in terms of how do you enhance that as opposed to the adaptive side of things. And again, we look a lot towards academic partnerships. I will say Memorial Sloan, MD Anderson, in doing a lot of that early stage work for us. So, I mean, wherever there's the unmet need, I mean, at the end of the day, you'd mentioned that's where you drive for is you're looking at the patient population. Where's the unmet need? And then what's available? I mean, at the end, and the one other thing that I would say is that there's less available in the market on late-stage clinical assets, and when they are available, you will pay for them. Mm-hmm. So, I mean, the premiums are still there. That has not dissipated.
Brian, do you want to weigh in?
Yeah, no, I agree. I think we're seeing a shift in investment to other modalities besides kind of the standard small molecule. And the last panel was even talking about approaches like PROTACs, like molecular glues that are kind of stretching the boundaries of what small molecules are. I think it was brought up before, cold tumors, how do you make them hot? I mean, I worked on the early CTLA-4, the first IO therapies. It was because there was an immunology group next door to a cancer group, in Groton, and Jim Allison came in and gave an amazing talk about the potential of this. Nowadays, we have bispecific IO molecules. We have T-cell engagers that probably might be a way in, right? A targeted way in, including intracellular targets with neoantigens for that to see if we can activate that immune environment. And then obviously we've talked a lot about ADCs. I think that cell therapies, absolutely. If we can get them into the solid tumor. Jose Baselga, who led our oncology research, that was his vision about how we can do that. We're seeing different ways in with armored T cells. I think having sort of the universal donor approach as well, buying the manufacturing, the complexity of the science, the expertise. It's very challenging, but we're also seeing lots of progress in this area. And again, it's going to be the patients that benefit.
So Teresa, one thing we talked about in preparing were challenges in manufacturing of cell therapies and some of the more complicated products. Do you want to talk about how Takeda is approaching that?
No, I mean, I'm going to have to say, I'm always so fascinated by the cell therapies. I mean, to me, it is just sort of a miracle of science to be able to do what the scientists have been able to do there. But it is a tricky manufacturing process, particularly for the one that we're doing at Takeda, where we're cryopreserving the cells. So that then they're ready for shipment immediately off the shelf. Ridiculously complicated from the non-scientist perspective. So, we already have built out a manufacturing facility for all of the clinical supply, and then are ready for scale up in another site where we're building that out now so that it'll be ready. It takes a lot of advanced planning to make sure that you can do that, because there's a recognition that you don't want to launch when you don't have enough supply. That's been happening again and again and again. And one of the other things that we don't talk about as much when we're talking about manufacturing is the sustainability elements that I think are very important. So, at Takeda, we are now, neutral on carbon emissions, goal being net zero by 2040. And it is stunning to me to watch what that takes in order to make that happen. Because when you look at your emissions across your manufacturing network, you control about 20-30%. The balance is coming through your suppliers. So now all of a sudden you have to work with the suppliers in addition to making sure that your entire network is reverting more towards, natural resources, et cetera, and then the packaging and how you do that. And it's something to also think about, that when we're looking at not only how do we do this complex manufacturing for the cell therapy, but how do we make sure that this is done in a sustainable way so we're going to hit that 2040 goal of net zero, which I think I think we probably have one of the most aggressive targets out there, but realizing you don't control a good 60 or 70% of that's coming through your suppliers.
Well, and I guess the other element of sustainability is the cost of the manufacture and some of these products, and are they actually going to be marketable based on the manufacturing costs? And there are a lot of technologies that are being developed now to decrease those costs, whether it's through optimizing the cells so that they're more productive, they're more stable. I guess I'd ask maybe Fred and Ushani, are you looking at those sorts of technologies that are looking to improve upon the manufacture of some of these new technologies?
So about a year and a half ago, we started a company called Ascend, which is a complex biologics manufacturer initially focused on AAV. Because Cancer Research UK had a site in North London that they didn't want to use anymore, and so they gave us the keys. And now we've got an additional site in Munich that came from Freeline and a process development group in Alameda in the Bay Area, and we're looking at adding to that. If you're talking about AAV as a gene therapy, the thing that we appreciate is that if you can decrease the empty capsid fraction and if you can improve the potency, you can take the costs down substantially. So the key to taking costs down is not necessarily making something more efficient where you still need to deliver 10 to the 12th or 10 to the 13th viral genomes per ml. The better answer is to have something that's 1,000-fold more potent so that in one run you probably could make enough for the planet. There are ways to move to suspension systems which will help manufacturing. And so for these very complex biologics, of which gene therapy and cell therapy are certainly in that bucket, I think that you need a process innovation group. One of the challenges of most cell and gene therapy companies that decide they should do their own manufacturing is that it's hard to have black belts in manufacturing work well with black belts in R&D. The R&D culture is I want to do a different experiment every day. The manufacturing culture is I want to do the same experiment every day. They don't tend to get on very well. The manufacturing guys say, I got to deliver this by the end of the quarter. The R&D guys say, I just have to have this product in 10 years. So, and yet for complex biologics, it's not like small molecules where you can say to some contract manufacturer, I just throw it over the wall, send me back the mass spec and the MR and the NMR scan and trace, and I'll know you made what I thought you were supposed to make. So the interaction between R&D and manufacturing is tighter, but they're different cats. And so because of that, I think building these complex manufacturers that can interact well with the R&D folks will be something that's important. I mean, MPM is doing that with Elevate. Arch did that with Resilience. It's a little easier to build these companies now because the assets are like 1/10 the price that they were a few years ago.
Yeah, and maybe to build on that, I think our view on maybe this challenge is maybe threefold, or three solutions that we've actually defined. The first, as Fred pointed to, was just the key bottleneck on manufacturing, and Elevate was our solution for that. So how do you remove that bottleneck from the process? I would say when Elevate launched, there was a massive amount of pull on very limited resources around manufacturing, because cell therapy was competing with the AAVs, lentivirals, and when you looked at established groups like Lonza's and so on, they didn't have slots available for your product. You tended to be on a waiting list for almost 2 years sometimes. And so the Elevate solution is, how do we solve for that simple step, which is having available space to scale and manufacture your product? The second solution that we've invested in is just removing manufacturing away from the process entirely. And that's with in vivo CAR. So we've made 2 bets in the space, one that's viral in nature, so that's Umoja, and the second one is non-viral, which is Orna. So Orna's solution, it's an LNP delivering an RNA where you can actually transfect immune cells and pretty much make the patient a factory for leveraging their endogenous immune system to create the engineered product. So what that does, it removes lymphodepletion, and it also removes the hurdle of manufacturing. Umoja has a similar kind of bet. They're specifically targeting T cells, with their viral vector, and again, also engineering the patient. So that removes manufacturing from the entire process, but these are still very early technologies. They haven't really gotten to the clinic. Umoja, for the first time, showed that you can actually have really good expansion of the transfected cells in vivo, and that's some NHP data that was announced publicly recently. And then the third area that we're starting to define a thesis around is how do you make the process faster, cheaper, better? We have one technology. It's an off-the-shelf iPSC-derived platform. So iPSCs are great. They're easily editable. They're easily scalable. They're consistent. You don't have batch-to-batch variation. But they're still quite challenging when it comes to creating a functional cell that is as potent as a non-iPSC. And so there are certain technologies that we started to see that gets you to the potency, gets you the scale. So currently, I think the best that we're seeing in the market is maybe 700 doses from a single clone. I think this technology potentially can give up to 15,000 doses. So that massively solves that scaling issue that we've been talking about. Then the process itself for getting to that scale is quite simple. So instead of having 3 steps where you have a feeder system, you have purification, you have digestion steps, you remove all of that with this specific technology and you have a single step that gets you to your end product. And so the simplification and the scale that this technology offers is why it's appealing, because we still do believe that some of the best responses that we're seeing in oncology is with cell therapy. We're not seeing massive amounts of CRs and cures and durability. I mean, I still love small molecules and antibodies, but it's not at the same level that we're seeing. We're not seeing the level of cures or potential cures, depending on how you define cures, with other modalities as you're seeing with cell therapy.
Well, hearing all these problems with manufacturing, I just want to turn this back to, I agree that cell therapy is the future, but there's, I think, many things that can be done with the existing technologies that are not being done because of the lack of good science or translational medicine in the process. And, you know, we mentioned this American Society of Clinical Oncology meeting. I probably could count on one hand the number of studies that are showing positive results in a refractory patient to immune therapy. Either patients who don't respond at all, and there's still many, despite the commercials we see on TV, many patients with lung cancer in my area still don't benefit, and more than half, if not 2/3, become resistant. So I would contest that, yes, let's work on the cell therapy, but there are antibodies, there are probably small molecules. Let's personalize the immunotherapy world, and it always, I can't understand why more of that's not being done. You know, so we give a PD-1 or a PD-1 inhibitor to all, and there might be 15, 20, 30 of these, but the real problem is going to be what is going on in that specific patient. Is it a hot tumor, a cold tumor? Are there resistance mechanisms in play? So, you know, given what we heard a little bit from Brian, we have to start thinking about what are the translational aspects we can put in place, maybe to do more, you know, some of our trials to be more scientifically based, maybe more adaptive, you know, to try to bring some of these therapies. Because I wonder if so many of these things fail because they don't get that phase 2 result. Because they might have 20% of patients might benefit. There's a signal there, but it's probably, you could probably get it up to 80% if we knew exactly what population that was.
So this whole concept of making cold tumors hotter, I think, is a really important one. To me, the biggest risk to these developments is a very pragmatic one, and that is that almost all of these agents are going to need to be used in combination with an anti-PD-1. And when you look for single agent activity in these drugs, which everybody seems to want to see, you often don't see it. And then there have been a lot of combination studies in phase 2 that look quite promising, only to see the phase 3 be flatline. And so I think, you know, my friends at senior levels in pharma are sitting there and saying, oh my God, the last thing I want to spend on is an IO agent that doesn't have clear single agent activity, but that looks like it could be used in combination based on this phase 2 data, you know, and I can blow a hole in $300 million and have a failed phase 3. And there've been a bunch of those, right? And so, I mean, you tell me if I'm wrong, but, right, but, you know, nobody likes playing goalie for the dark team, and that's the way I think people feel in all of this. I mean, I guess it's better than playing catcher for the javelin team, right? But no, I mean, it's been hard. And yet it's clear that that happens. And if you were to look at the biopsies, you see immune cells kind of hanging out nearby but not getting in, which is telling you that there's some signal, tumors are clever, that they're putting out that's saying, oh, you've come close enough, right? We think we know what some of those agents are, but we haven't quite figured out either what the right agents are or how to get those agents into that zone. I was listening to somebody tell me that because of the tortured vasculature that tumors recruit, it's actually a higher pressure system. And so viscous agents don't flow there.
Judah Folkman was talking about that 25 years ago, and he was right. And I would contest that even the cell therapies, they probably won't work in all patients for the very mechanism you just realized. You can put all the T cells you want in, but if those genetic mechanisms from the tumor are keeping the TILs out, and you're right, we've seen this in our own work here, we've published it, a rim of T cells around the tumor, they just don't get in. That's an area where I would look for companies that are doing that sort of work, get the T cells into the tumor. But again, it's hard because these are complicated trials. It's, to get a biopsy and use that biopsy, it's not easy, but it's the type of thing I think we have to do maybe as a public-private partnership, all coming together to do that.
So I want to thank our panelists. I think we are up against time. We got through most of our topics, but thank you so much for all of your input. Really appreciate it.